Assalamu alaikum warahmatullahi wabarakatuh, and very good day to all of you, and hello. So today we are going to do practical number 5, which is about multiple sequence alignment. Multiple sequence alignment is collection of more than two protein or nucleic acid sequence that are partially or completed aligned. So it is used for located conserved region and non-conserved region that are used to locate distantly protein members that are have not related to each other, and for annotate motifs, active site, uh, pentapeptide, lead metal binding sites of a target of sequence. There are many steps and many ways to get a multiple sequence alignment by exact method or progress method or iterated method, also consistency based method or structure based method. So here we will do different by using different uh, algorithms and programs related to that steps, such as Kestrel, W, Paralion, and T-Coffee, Espresso, and so on. And at the end, in the report, we will compare between these steps and method to know which one is better and which one is provide information on this and doesn't. As we know, for example, for progressive methods by using Kestrel W, if there is a gap, it will always will be there's a gap. So let us start by doing practical 5.1, which is multiple sequence alignment using biology bench workbench interface. In this practical, we will do some that may be new for us, like using in number 3, perform signal B to locate signal peptide, and also we will go across something called C score and S score and Y score. And before we start, I want to explain something that will help us to understand all of this. Let us see this picture. So this is a cell membrane, and this is inside the cell, and this is in the black color is the nucleus that contains the DNA. In the red color, this is the protein. And as we know, is this part is at the beginning of this protein is a sequence of amino acid that called signal peptide. So signal peptide is used by cell to relocate this protein and reposition this protein in different cells. Signal peptide is as in the sequence it at the end it is come to mRNA, and in mRNA, as we see here, there is signal peptide sequence, and the rest is uh, the protein sequence, the protein coding sequence. So in this practical, actually we just wanna use protein sequence. So we wanna use the amino acid of that sequence for protein. So we don't need to go across the sequence and amino acid of signal peptide. So we need to remove this part from the other part of protein, because this is coming together, but there is a way and the steps to remove signal peptide sequence from uh, from the rest of protein, and we just use the rest of protein to do multiple sequence alignment. So since mRNA is transcribed from DNA also here, so here's the sequence of signal peptides that related and connected to the sequence of the protein. So signal B4, as I mentioned, Here and number three is a tool to locate signal peptide that related and connected to that protein. So we will remove it after we we want to know it. Why we need to remove signal peptide and just we want to keep the amino acid of protein because in this we want to make we want to make a multiple sequence alignment and this signal peptides is change from protein to different proteins, so we gonna use the one that is not doesn't change. And also just before we start is this signal peptide is at the beginning or the end because correspond and it will be N terminus or C terminus, but usually it will be N terminus, and we will see in this practical. So we will start now by, this is just introduction to get some knowledge. So now we will start by doing this. So the first step is going to NSPI. We will open Google NSPI. Okay. Second step that we will use the sequence that we have researched by GenBack accession number, such AY260764. Copy and paste. Here we see this gene. It is a lipase gene. It is a DNA sequence that is called G. bacillus zahila strain T1 thermostable lipase genes. So we go to FASTA this genes to get the sequence of this gene. So this is the sequence of uh, this DNA. So now we wanna go to perform signal B40 4.0. Just type in Google signal B 4.0. As we see here, there is signal signal B4, and there is a signal B5, which is the newest one. So here we will gonna use this one. This is the signal B5.0 server, and here we submit our sequence. But actually here this is used for protein, and the sequence that we get it's just for DNA. So we will use some other tool that transfer or translate DNA to protein. So I will type in Google translate DNA to protein. Translate tool, we will use ExBASy. It is one of the best. So here in this, I will paste this one I get. So I will copy, paste, and translate. Then this is the result that we get. As we see here, there is many frames that we get, around 6 frame, and 3 for each direction, 3 from 5 to 3 frame 1, 5 to 3 frame 2, 5 to 3 frame 3. And in the opposite, there is 3 to 5 frame 1, 3 to 5 frame 2, and 3 to 5 frame 3. And as I said in the beginning, we will focus on N terminals, which is 5 to 3 frame 1. So, and this is sequence of amino acids that we see. So we want to take the sequence of amino acid that related to protein. So we want to discover which part is related to protein and which part is related to signal peptide that we can remove. For example, if we take 5 to 3 frame 1, here there is, as we see, there is some region that is highlighted by a red color, like this one, until here, and another one is from starting codon until here. So I have a 5 region that is highlighted. So start by methanoids are usually used a lot at starting codon. So one of the hints that tell us which one is the protein that you are wanna use it for multiple and do multiple sequence alignment is which one is the longest one. So it is obvious that the longest one is from here to here. So just I will I will take this and I will highlight it, and I wanna make copy and paste. So now I highlighted the one that uh, because it is the biggest one, and this is the hint that we use to know which one is the suitable and which one is the better. So this one contains two parts, contain the amino acid and sequence of signal peptides and amino acid and sequence for protein. So now this one, after I take this and I copy this and I use it and I post it in signal P5.2. So as we said here, this is used to locate signal peptide that will tell us where is exactly the signal peptide to remove it. But instead, in using signal P4, I use it signal P5. So to recap, I take the sequence for the protein, but for the DNA, and I change it to sequence of proteins, and I take the longest one and the one that I will paste here, then submit. Now I'm processing the job and we are waiting for the result. Now we, and this is the result, the usually result that we get from signal P5.0. So uh, as I mentioned before, that we will go through something called S-score, S-score and C-score and other scores which we called here yellow score. So the red one, this is the S score, and the green one, this is the C score, and the yellow one, this is the Y score. And here is the protein sequence, the protein that we take it from XBC and translate in signal B. So this protein sequence is contain the signal peptide that we need to remove and the sequence of the protein that we need to make a multiple sequence alignment for it. So how we can know which from where this start and end, and the one start and end is using these three scores. So by starting by red score, what is S score? S score is starting at a high, but here start to go down. Once S start to go down, that tell us this is start at the end of the signal peptide. So from here until this point, before it's go down, this mean in this point, this is the end of the signal peptide and the start of the sequence of protein. So S square tell us until here, this from here until here, this is the signal peptide, and from here to here, this is the sequence of protein. For C score, the green one, is tell us at the highest point in this score is the starting point of protein. So we will look where is the highest point at the C. This is the highest score, and that tell us this is the beginning of the sequence of protein, and that means before it is the sequence of signal peptide, and it is the same with the red one, yellow one, red one. So S score, red one, and C score, green one, tell us it is the same. So that's tell us it is. This is the second hint uh, to locate the signal peptide. And the last one is the yellow score, which is yellow color, that give us a range from what are from down after when it start to going up until here. That's tell us the between this range is the signal peptides are ending and protein are starting, which mean the confirm ours by that we get two hints, and this one is confirm the region, so it will be here. So that's mean from 0 until 20 and 21, 22, 23, 24, 25, 26, 27. That is the last. This is until 27. This is the amino acids for signal peptide, and from 28 until the end, it is the amino acids and sequence for protein that we will use. So now we will go and remove all of these, and we will just take from 28 amino acids and until the end, that it will be the protein that we will make a sequence for lipase protein that we will make as multiple sequence alignment. So now I will go to XPC here, and I will count from 1 until 27, and I will remove that because that one is for signal peptide, and I will take the rest. But I already count until A is residue. A is the this is the 27. So from here until the end is the sequence for protein, and this one I will make. This is the mature lipase and the mature protein. So we'll come back to the manual. So after we perform signal 5 to locate signal peptide, we will go to blast B to we will go to blast B, and we will click the protein plus to paste the amino acid sequence for mature lipase in the window that we get after cut the sequence of signal peptide. I will click protein plus, and here I will paste this one that the highlighted one that I already copied. Oh, sorry, so I need to make sure it is blast B, then I click blast. We are waiting for the result. We get the result and we see whole, this is the multiple sequence that we get, and it is 89 sequence selected. So this is the identity between this. As we see, it is to start by 100 percent, and it is not below than 90. So it is the lowest one is around 93 and or 93 point 0.06. So we click the protein and we paste the amino acid for matchable and we look at and we get the sequence. But as we see it here in the practical, it asks to select a few sequence to each with the following identity, which is ask us to select two pairs that one pair is from 91 to 100 and one of, for example, 61 to 70 or 31 to 40. But in this proteins, the minimum one that we get for identity is above 80. So we will choose around six sequence that is between 100 to 91 instead of this because we don't have that. And when we choose, uh, we will select, we will try to make vary between them. So maybe the first two of the first one pair we choose 100 and the other one is 99. This is the first pair, so but we will make little difference between them. Maybe now we will say 96 for identity, and we will, this is 96 for 40, and we will choose 97. And the third pair, maybe we'll choose between 90, 30 and 92. In this, we we will get now the multiple sequence alignment. We make a download. We can choose for faster or a text, anyone that we can will start faster. And here finally uh, we get the result, and this is the multiple choice, and this is the multiple choice uh, for this protein that we, and this multiple choice we will use it for. Now we came to the end of practical. At this point it is the end of practical 5.1. We get the multiple sequence alignment, and that multiple sequence alignment, it will be used for practical 5.2, and the same itself, it will be used at practical 5.3, and it is practical 5.4. And actually we almost done the majority part of the practical, since the others is just uh, click the paste the sequence faster format and get the result. So now I will start by practical 5.2.